My name is Fabiha Anwar,
And I'm Yusra Alam.
and today we're going to be going over a study done by Zhengna Zhang, who covers systematic analyses reveal uniqueness and origin of the CFEM domain in fungi. And some keywords from this paper include CFEM domain, which is a component in fungal proteins or a sequence of nucleotides that distinguish fungi and cause toxicity. Basidiomycota is a phylum in the kingdom fungi. Genomic analysis is identifying and sequencing DNA, and in this case, we'll specifically be using CFEM. EST, or express sequence tags. They detect the presence of certain sequences of nucleotides. In this case, we will be using CFEM. PCR, or polymerase chain reaction, which amplifies specific gene sequences, which can be used in gel electrophoresis.
Zhang's study was conducted in 2015, and its purpose was to figure out where CFEM fell in the phylogenetic tree for kingdom fungi. It was first seen in adenylate cyclase as well as the rice blast disease. But CFEM was noted to have different functions in different genuses of the kingdom fungi. For instance, Zhang and his team took a look at the genus Saccharomyces, part of the phylum Ascomycota, and noted that CFEM present actually helped more in structural support than toxicity. However, on the other hand, CFEM can have toxic traits in the rest of Ascomycota and Basidiomycota.
There are several competing hypotheses for how CFEM originated. For example, one of these hypotheses is that CFEM arose independently in the phylums Basidiomycota, Ascomycota, the subphylum Saccharomycontina, and Pizizomycontina. However, Zhang actually hypothesized that CFEM originated in a common ancestor of Basidiomycota and Ascomycota, given that it's only absent in one Ascomycota lineage. And the loss of CFEM is more plausible than independent evolution in several different phylums. Materials and methodology. Experiment 1. Zea mays and sorghum bicolor, which are two plants, were used in this experiment. Their seeds were sterilized using 75% alcohol and 0.01% mercury chloride. The cotyledons were extracted from these seeds, calloused, and were grown in a sterile environment. The control of this experiment also used Zea mays and sorghum bicolor. However, in this case, the seeds were not sterilized. Their cotyledons were simply extracted, calloused, and cultured. PCR was used to amplify CFEM as well as RNA and DNA from both of these seed cultures. Primers developed from ESTs were used to identify the presence of CFEM in both our control, the unsterilized tissue, and our experimental tissue, the sterilized tissue.
In the second portion of the experiment, Zhang used an online database, PFAM, to get fungal genetic sequences and compare them to the CFEM sequence. Those that matched CFEM closest were then plotted onto a phylogenetic tree.
Results. Experiment 1. In figure 1, we can see a picture of the calluses from Zea may and S bicolor. And in part 2, we can see the gel electrophoresis, which uses CFEM as a marker and compares it to the plant tissues taken from both plants. In the unsterilized callus sample, CFEM is present. However, in the sterilized sample, CFEM is not present. To explain these results, Zhang hypothesized that the plants could have obtained CFEM via lateral gene transfer, which seemed unlikely, or that the genes that sequence CFEM were just not caught in the gel electrophoresis for both plants, which also seemed unlikely. Zhang hypothesized the presence of CFEM in the unsterilized plant tissue actually came from an endophytic fungi living in both plants. This hypothesis is supported considering that CFEM was not found in the sterilized plant tissue.
In the results for experiment 2, a phylogenetic tree was created with the results of the CFEM compared to the fungal genes. It was seen that the phyla Microsporidia, Chytridiomycota, and Zygomycota had little to no presence of CFEM, whereas Ascomycota and Basidiomycota had high levels of CFEM in them, indicating toxicity.
Discussion. Experiment 1 concluded that CFEM is mostly found in fungi. And if it's found in other organisms, CFEM may be present but has other purposes. For example, we looked at CFEM being found in the endophytic fungi rather than the plant. CFEM here is primarily causing toxicity. This endophytic fungi actually preys upon the plants that it was found in. And this results from experiment 1 was later used to complete the phylogenetic tree in experiment 2.
In experiment 2, Zhang and his team discovered that CFEM evolved from the common ancestor of Ascomycota and Basidiomycota, denoted by the star in figure 2. This is most logical because it makes sense for CFEM to evolve from Basidiomycota and Ascomycota, only lose the trait in classes like Taphragnomycotina. Furthermore, it makes sense that the pathogenic owed most of its toxicity to CFEM. Works from Zhang and his team connect to Bio 1108, specifically Chapter 29, where we learn about Basidiomycota and how it's toxic and is harmful to organisms that consume it. This is supported by the phylogenetic tree, where all of Basidiomycota is red, indicating levels of CFEM, and thus they are pathogenic.
Zhang's work is imperative to truly understanding previous CFEM research. I want to take a moment and look at previous research that was done by Dr. Schaefer in 1989, where he actually hypothesized that there was a domain in fungi that made certain fungi pathogenic, and not necessarily saprophytic. It was Zhang's study that helps clarify that this hypothesized domain was actually CFEM. There is certainly much more work to be done on differentiating certain phylums in the phylogenetic tree of fungi. Zhang's work is only one of the many missing pieces.